This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
Since anthropogeny is not a normally common word, although many people are getting to know it, I hope, it is the investigation of the origin of humans, and we do that writ large. And I think you'll see today that what we're talking about today is not the actual depth of origin of modern humans, but it does have implications for how that came about. Our complete CARTA mission statement, which we've worked on, is to use all rational and ethical approaches to all verifiable facts from all relevant disciplines, and you can tell just from us there are a lot of relevant disciplines, to explore and explain the origins of the human phenomena. We try to minimize complex organizational structures and hierarchies, we try as best we can, and to avoid unnecessary paperwork and bureaucracy. So we try to keep it simple. I do want to thank some people because without them, we wouldn't have CARTA and we wouldn't have these symposia that we've been able to carry on. Our main supporter is the Harold and Lila Mathers Foundation, and I would especially like to thank Mr. Jim Handelman, the executive director, and his wife Susan. I'd also like to thank Annette Merle-Smith, who could not be with us today. Now, I will turn this over to the co-chair of today's symposium, Dr. Evan Eichler from the University of Washington.
So it's a pleasure to welcome all of you here today. I want a special thanks to the speakers that have come from far and wide to make this symposium an event. We came up with this idea about 9 months ago, and I can tell you that it came from really a mixture of new data that was emerging, the enthusiasm from complete sequencing of genomes, and the fact that it's been almost 10 years since we've had a human genome reference, the ability to start to kind of peer into the diversity that exists in different continental groups, and to understand that really from a functional perspective is why we're all gathered here today. So this symposium is organized in two parts. The first half will be really kind of exploring the nature of genetic diversity in different continental groups. And we're going to hear some excellent lectures focusing on Africa, South America, and Europe, and what we've learned about human genetic diversity just in terms of neutral genetic diversity and its implications. We're also going to hear from Pascal Gagnon regarding kind of where we fit with respect to other ape species or other species related to humans. And I think he'll leave you with the impression that we're quite shallow genetically as a species. He'll also give us a little bit of a primer in terms of, so everybody's on the same page in terms of what we mean when we talk about genetic variation. The second half of the symposium will focus more on really some of the functional implications in terms of brain development, in terms of immune response, and we'll end with some discussion regarding the impact this might have in terms of society. Our next speaker is Carlos Bustamante. He's now at Stanford. He started his lab there in the beginning of this year. He graduated in 2001 with a PhD from Harvard in biology, and from 2002 to 2008, he really moved up the ranks very quickly, rising to full professor status in a very short order, in large part due to his accomplishments that he's had, particularly in the area of genetic diversity. He has a wide range of interests that go from all the way from dogs to humans. And I think we're going to hear mainly about his work with European diversity, but I think he'll dabble in other areas as well.
I'd like to begin by hearkening back to what Evan was saying about the technological developments that have arisen since the International Haplotype Map Project and other large sequencing endeavors. So as many of you know, these technological developments have now led to a 100 fold, if not more, sort of cost in the reduction in the cost of genotyping. So I used to have a slide on here as to how much it would cost to genotype 1 million variants in the human genome, but it's now gotten to the point that you just need to update it every week because it just becomes cheaper and cheaper and cheaper. And the DNA sequencing has gotten to the same point with complete human genomes. So if we think about the collection of the first complete human genome sequence, it took about 20 years and on the order of $1 billion. The second one took several $100 million, and then from then on it's just dropped precipitously. We're probably on the range of about $5,000 to $10,000 to sequence a human genome today. Probably at the end of the year that'll be about an order of magnitude less. So we're really sort of entering this realm in which sort of collecting full genome sequence data is really no longer sort of the barrier to thinking about problems, but rather really sort of thinking about the biological and medical problem that we're interested in. And as Evan was saying, sort of in my lab alone, we've had the tremendous pleasure to work on a wide range of organisms ranging from humans to dog to rice to cattle. And what has made this sort of fun and exciting is that oftentimes we're interested in the same set of questions. So we'd like to quantify genetic differences within and among populations, a very classic problem in population genetics. We'd like to identify the genes underlying traits of interest. So we heard a beautiful study today from Sarah about mapping mutations involved in lactase persistence. In the same way, we'd like to use genome-wide studies to understand genes contributing to disease susceptibility in humans, or morphology in dogs, or milk yield in cattle. Likewise, we'd like to reconstruct the demographic history of different populations and identify targets of natural selection, again, akin to what Sarah talked about today, but really sort of on a whole genome-wide scale, looking at patterns of variation for many different populations. And in particular, I'd like to focus today on the problem of personal genomic sequence and trying to understand what is that we can glean from genotype data and eventually sequence data from about personal ancestry and differences within and among populations. And as a way of motivating this, I've put a figure on here from one of the largest studies to date in medical genomics, namely the Wellcome Trust Case Control Study, which aimed to identify genetic variants associated with 7 different diseases simultaneously. So this is really sort of a paragon of what is being done today in medical genetics. And this was a study led by Peter Donnelly and others. And they, as I said, they looked at bipolar disorder, coronary heart disease, a series of different complex diseases. These are so-called Manhattan plots. Each of these is a chromosome, and the height of these bars is giving you a sense of the statistical evidence for associations among genetic variants in a region and a disease. So for example, there's a hit on chromosome 9 for coronary heart disease. There's a bunch of stuff for Crohn's disease all over the genome. They really didn't find anything for hypertension and so on. One of the main limitations to this sort of research at the moment is that these studies are being done in a small corner of human genomic diversity. In particular, most of the individuals that are being enrolled in these sorts of studies are individuals of Northern European descent. So, and there's now a move to study a host of different disorders, glaucoma, ischemic stroke, and so on. And a huge question is, what is the relevance of the variants that are being found by these studies to other populations? And we heard, again, another wonderful example from Sarah, where the mutation that causes lactase persistence in Africans is quite different than the mutation that it's in the same gene, but it's a different variant than you find in European. So it's a question of how common is this a picture, and how do we really enable a medical genomics on a global scale? And so to answer these questions, we really need to take a look at classical questions in human population genetics. That is, to what degree are modern human populations accessible for medical genetic studies substructured? That is, if I'm running a hospital in Kinshasa or in Caracas or in Karachi, how different are the people that are coming into my clinic, and how can I use that information to design a carefully controlled genetic studies? Second, what are accurate models for describing human genomic diversity? And what is it that we're learning from the latest sort of technologies, both genotyping and sequencing? How can we make use of these data to reconstruct personal genetic history? That is, are we able to learn about the history of our genomes, about those different segments that Pascal was telling us about today from looking at these sorts of data? how do we think about reconstructing ancestry, and will this be necessary and sufficient to allow for mapping of disease mutations across divergent populations? How do we think about, again, this question of enabling a medical genomics on a large scale? And how do we undertake genome-wide association mapping in admixed populations with complex structure? And we'll talk about that in a moment. So the study I'd like to tell you about today is a sort of long-standing collaboration between my lab and GlaxoSmithKline. I don't have any stock in Glaxo, they don't pay me. It just happened to be a collaboration that we had. And the study aimed to establish a resource for studying human population genetics, recent demography, and admixture. We spent several years dealing with this bit, which is trying to get all the data together. I won't bore you with the details. I'll just leave you with this number, which is that when we took two individuals and we genotyped them, 99.5% of the time, we had the exact same genotype for the position. When we started, it was not nearly as good, and we'll leave it at that. So the study enrolled about 5,000 individuals, and this was really one of the largest studies on this scale that looked at global genomic diversity. And in particular, we had 3,200 Europeans from most countries in Europe, with hundreds of individuals per country, or at least dozens of individuals per country. We genotyped 500 African Americans across 500,000 markers, one of the largest studies to enroll African Americans, and we'll talk about patterns of genomic diversity in African Americans. With Sarah, we looked at 200 West Africans that I'll also discuss, Indian Asians, which until this point had never been looked at that scale, as well as East Asians and Mexicans. And I apologize, there was a problem with transferring the file, but hopefully this will go through. And as I said, Matt Nelson led this effort at GSK, and our collaborator, John November at UCLA. So one of the big questions in trying to think about these data is that the SNPs that you're looking at, that is those variable nucleotides, aren't random, right? They are sort of drawn from a bizarre kind of constellation of the possible SNPs one could have looked at, and it turns out they tend to favor common variants. And the other issue is that there's a huge amount of data. So if you imagine an Excel spreadsheet, you'd have about 500,000 columns and something like 5,000 rows. There's a huge amount of data, and you can imagine putting a clever undergraduate to look at each of the columns, but they'd soon sort of get bored and go on to a different lab. So we really need to think about ways of grappling with the sort of just magnitude of data. And what I'd like to tell you about is an approach that we've found particularly useful, something called principal components analysis. And I think the easiest way of thinking about principal component analysis is a way of visualizing 500,000 dimensions at the same time. So if you imagine just 2 dimensions, you could imagine that you might have data that's sort of correlated along this XY axis. What principal component would do is simply treat this as the new axis. So that would be here, this would be the diagonal, and then tell you there's not much going on in the off diagonal. The nice thing about PCA is that if you have clusters in this large 500,000 dimensional space, it will retain that structure and really sort of make that clear. Okay, so now on to looking at 500,000 dimensions of human genetic diversity simultaneously. And when you do this, you see that the patterns that emerge actually are quite interpretable. So each dot here is an individual, and I've colored them according to self-reported ancestry. And what you find here, we're looking at those first 2 dimensions, the first 2 most important dimensions of that 500,000 dimensional space. And you see that it corresponds extraordinarily closely to the Africa out of Africa axis. That is, here are the individuals from Nigeria, the Yoruba, West Africans. Here are individuals from Europe and East Asia along principal component 1. Here are individuals of African American ancestry, and you see that they are on a continuum along this axis, and we'll return to this point. This reflects, as Sarah mentioned, the admixed history of these populations. Principal component 2 separates the East Asians from the Europeans, and you have the South Asians, so the individuals from India and Pakistan in between. On the other side of the globe, you have individuals with ancestry from Mexico, and presumably what you're picking up on is a signal of admixture that comes from Native American and European ancestry. The third principal component separates the South Asians from the East Asians. And the fourth is one we were particularly interested in, one that is only impacting the Mexican sample and is really essentially separating individuals of Native American ancestry from those of East Asia, even though we didn't have Native Americans in the sample. What you're really looking at here is a signature of Mexican admixture and the admixed ancestry of these populations. So you really begin to see that these genetic data have a fair amount of information regarding ancestry. And if we intersect this with one of the largest studies to date to look at global patterns of diversity, a study by Stanford on a panel called the Human Genome Diversity Panel, you begin to see this sort of beautiful picture, a sort of mosaic of how different populations are related each to the other, and how each human population is essentially one end on a continuum of lots of different human populations. And in fact, this notion that you might get from just looking at 3 or 4 geographically distinct populations of little clusters is really just an artifact of your sampling. And in fact, we have this sort of bleeding of one population into the other. Here's, for example, the Mexican admixture signal, where you have Native Americans, here are Europeans, and this sort of the continuum of individuals with admixed ancestry. So we really are doing ourselves a disservice by restricting medical genomics to this sort of small corner of genetic diversity space. So you might say, well, Carlos, we need to start somewhere. Europeans, they seem to be a sort of monolithic group. Perhaps that's a good place to start. And so let's just focus in on this aspect of the map and PCA that up. And what you find is that, oh, this is unfortunate that this transferred poorly, but what you see here is that you get a map of Europe from PCA that reflects the geographic map of Europe. So here are the Spanish and the Portuguese on the Iberian Peninsula. Here are the Italians. These individuals actually are from Sicily. Here's the UK, the US and the UK and their special relationship. Here's the Greeks and the Turks near each other, which is good for us, not necessarily good for the Greeks and the Turks. So you begin to see this sort of really sort of reconstructing of physical distance from genetic distance. And you might say, well, Carlos, all right, fine, let's just focus in on Switzerland. I know that's where Pascal is from. He might be interested in this. So if we do that, you see that even within Switzerland, you have all kinds of population substructure. So the French-speaking Swiss cluster with the French, the German-speaking Swiss with the Germans, and the Italian-speaking Swiss with the Italians. So there is a huge amount of genetic diversity within the human species, and this genetic diversity is really reflecting patterns of migrations and patterns of non-random mating, and these technologies are giving us deep insights into this, and this is something that we really need to first understand, secondly, sort of grapple with, and third, really kind of interpret well, for the reason that this is just going to become ever more precise. So as I said, we're focusing on patterns from common genetic variants. Now that we're beginning to sequence, you're going to begin to look at rare genetic variants, and those rare genetic variants are likely to be even more diagnostic of ancestry. So you might, for example, only find a variant in a small valley, one of the small valleys in Switzerland, and so if you carry that variant for a certain segment of your genome, you are almost surely has descended from that sort of small valley. In this way, we may be able to sort of patchwork and understand from the genetics, the sort of personal genetic ancestry. So I've always loved this painting from Norman Rockwell, where little this boy knows the sort of richness of his ancestry. He has ancestry from Native Americans, perhaps a pair of brothers who fought in the Civil War, and then trace their ancestry back to Barbary pirates that landed off the coast of Florida. So can we get this sort of information? How well can we reconstruct these kinds of patterns? And our first stab at this was in the November et al. Paper that was published in Nature, which took the PCA data and used a very sort of simple model to try to predict longitude and latitude from the first 2 principal components. And what you see is that you do sort of surprisingly well. So here are the individuals that you predict to be from Spain. Here are the individuals from Italy, from Portugal, and where they actually arise from. And the accuracy is something like 50% of individuals are predicted from within 400 kilometers of where they were born. And these are individuals that have 4 grandparents from a given country. So there's a tremendous amount of precision. And as I said, as we start sequencing, we're only going to improve on this kind of ability to predict ancestry from genetic data. I'd like now to shift focus and talk a little bit about patterns of genetic variation in African Americans and West Africans. This is, as I said, a collaboration with Sarah Tishkoff and a paper we published earlier this year in PNAS. And the idea was essentially to think about each of these individuals and try to estimate genome-wide the ancestry components that came from each of the different potential source populations. And as you know, there's a tremendous interest within the African American community for solving this kind of problem. Due to the barbarisms of the transatlantic slave trade, they've lost a huge amount of their history, and they'd love to understand, can you take segments of my genome and assign them to a particular population in Africa, a particular population in Europe, and so on. And this is the work of Kasia Brick, a just outstanding graduate student in my lab. And what Kasia sort of hit upon is the idea that perhaps the distances in PCA space themselves may be good proxies for ancestry. So you might imagine genome-wide, you would estimate at this point the ancestry is just a linear distance of the relative distance of from the European centroid relative to the West African centroid. And it turns out that that does extraordinarily well and is very well correlated with this measure that Sarah talked about, the structure program. But what's neat about Kasia's approach is that you can actually run it genome-wide. So for example, in blue, here's the average of PC1 score for all Africans in the data set. In red is the average of PC1 score for all Europeans in the data set. And in black, here's a single African American individual. And you see that they almost look like signals coming from a channel. And this individual sort of right in between the blue channel and the red channel. So you might sort of deduce from this that they have one chromosome of recent African origin and one chromosome of recent European origin. This is all along chromosome 1. Here is another individual along chromosome 1. And you see for the first half, they trace the same as this first individual. But now there sort of seems to be a switch. And they now look to be tracing the two African chromosome segments. So you would deduce here that there is now been an ancestry switch in this individual. In this way, you can overlay a mathematical model called the hidden Markov model. And here's what the raw data looks like, the sort of jagged jumpiness. And then you put that through the model, and the model makes predictions. Here are two chromosomes of African origin. Here's a region of European African origin, and you get these sort of beautiful oscillations in the data which reflect the personal genetic ancestry of this individual. And in fact, the length of those segments are telling you about the particular admixture history of that individual. And here are sort of four chromosomes, four genome-wide reconstructions of ancestry from 4 different African Americans in the data set. So here, for example, is a representative African American. Blue is a region of African ancestry. Green's a region of joint European African ancestry. And red is a region of European ancestry. Here's an individual that seems to have had a more recent admixture history because they seem to have longer segments of African or European ancestry. Here's an individual that only seems to have European and African ancestry or European ancestry, which suggests that they have one parent of European origin. And here's an individual who's self-identified as African American, but actually seems to be largely of European ancestry. So these are sort of representative of the sorts of data that we have obtained, and we're now in the process of figuring out how well you can assign these segments to different populations. And one of the things that you find is that, in fact, it turns out to be quite difficult, and even after you've looked at 400,000 or 500,000 markers, particularly within West Africa. And that could be for two potential reasons. One, SNP discovery in West African populations has not been properly done, so we haven't sequenced enough West African populations to identify markers that can delineate ancestry. Or two, there was a sort of complex admixture and migrations at the time of the founding of African American populations. So what you really have are the coming together of several populations right at the time of the transatlantic slave trade. Okay, I'd like to just finish by telling you a little bit of the story from Latin America. I happen to be from Venezuela, so we've been collaborating now with folks to look at patterns of genomic diversity in the Americas. And what's particularly interesting here, so we've got samples from Puerto Rico, from the Dominican Republic. As you can see here, it shares half of Hispaniola with Haiti, from Colombia, Ecuador, and Mexico. And when you look at patterns of Hispanic Latino diversity, you see that they're sort of all over the place. So just to orient ourselves on the map here, here's the cluster of European ancestry. Here's a cluster of West Africans. Here's the cluster of Native Americans. And here, each of these represents samples from a different country. So individuals from Mexico are primarily on a European Native American axis of ancestry. individuals from Colombia seem to sort of be in this space. They have sort of three-way admixture. From Puerto Rico, a slightly more three-way admixture with less Native American ancestry. And here individuals from the Dominican Republic, which you see essentially look very similar to African Americans in that they are on a European West African axis of diversity, except it's closer maybe to a 50-50 ancestry as opposed to 2080. And this is particularly interesting in the case of the Dominican Republic for the reason that they have historically had a national view of themselves as derived from Taino Indians, which, and in fact, there's a fair amount of discrimination in the Dominican Republic against Haitians. But in fact, they really have an African admixed ancestry, and patterns are different between the European, the autosome and X chromosome, and we can talk about that as folks are interested. With that, I'd like to just to summarize and say that new technologies provide fine-scale genome-wide view of population structure and admixture. It's important to realize that these results reinforce the prevailing view since the 70s that human population structure is largely spatial and clinal. There is sort of no single sort of monolithic clustering by race or any kind of notion such as that. In fact, it's all sort of spatial and clinal. We need to embrace admixture. It's a challenge for genome-wide association mapping, and this sort of personal ancestry reconstruction is quite feasible on a genome-wide scale. And thank you very much.
My name is Ajit Varki, and as the co-director of UCSD Salk CARTA, it's my great pleasure to make a few closing remarks to this wonderful symposium. I would obviously like to thank the chairs and all the speakers for a truly intriguing and thought-provoking series of presentations. And of course, special thanks to our major sponsors, the Harold and Lila Mathers Charitable Foundation, and Jim Handelman, the executive director, and Annette Merle-Smith. This afternoon, we've been treated to a fascinating feast of current information on the origin of the human species and our remarkable biodiversity. So it's said that truth is stranger than fiction. Who would have thought that all of us in this room, and in fact all of us on this planet, came from less than 5 or 10,000 individuals less than 100,000 years ago? So even while we celebrate the rich diversity of humans, I think we should, with Svante Pääbo, realize that from the genomic perspective, we are all Africans, either living in Africa or in quite recent exile from Africa. So in this regard, I cannot help but close and mention something that I came across last week, and that was a reminder about the upcoming US Census. Question number 9 on the census form asked me to classify myself into this bewildering set of races. And although I'm technically an Asian Indian, as you just heard from Mike, that's really not a very good category. In fact, the part of India that I'm from has not even been tested. So my own ancestry probably involves ancient Dravidians, Indo-European invaders, immigrants from Syria, Chinese sailors, Arab traders, and who knows who else. So I feel that I must instead check the box other. But then I have to make two choices. My first choice is my true ethnic identity, which is culturally westernized, anglicized, naturalized US immigrant, Syrian Christian Malayali from Kerala and the south coast of West India. So I thought about this, and I think what I'm going to do instead, and I would encourage you to tell everyone to do this, let's make a change here and just say human.